Hello, everybody out there in Microland. It's time for recorded asynchronous lecture 10 for Bio 145 Microbiology. This lecture is scheduled for Friday, February 6th of the year 2026, and today's topic is a continuation of industrial microbiology. Let's do the recap. So we were just saying that many industrial microbiology products are natural metabolites. These are microbes that have not been in any way genetically modified. You go back thousands of years and these microbes will be making these things, all right? And just to remind you that primary metabolites are those that are made during trophophase, meaning they're associated when the microbes are growing well and heartily and happily. Usually they're things that are made by the microbe to give to their daughter cells. Okay. Secondary metabolites are made during idiophase. Idiophase is like thinking like stationary or even decline phase sometimes. They're made when the microbes are not growing well. Okay. And those are kind of weirder things, from antibiotics to things like hallucinogens even. No one knows sometimes why the microbes even make them. And these industrial products, usually they're made, industrially at large scale. So we say these containers or these vessels that hold the microbes, and sometimes they be thousands of liters, hundreds of thousands of liters. We call them fermenters, not because they're doing fermentation, they're just growing up in large scale. And you can call that fermentation too, and I know most of you think, well, fermentation, that's when NADH is dropping its electrons on some organic molecule. Well, fermentation can also mean just large scale growth of microbes to make some industrial product. And here are just the pictures just to remind you, a primary metabolite is made during primary phase while as the microbe grows well. A secondary metabolite is made after the microbe has grown well. It's made in idiophase, all right? So the cells really aren't increasing anymore. They're making it after the cells have kind of almost stopped really growing. They're still growing, but not as well. All right, so let's now go into the new stuff. Welcome to part 2. So we were talking about what are some natural products of industrial microbiology that are out there. And I think you've seen a lot of these. Number one, certain chemicals like alcohols, all right, amino acids that you buy in the health food store like lysine, or amino acids for flavoring. Yeah, monosodium glutamate is glutamate. It's lactic acid there, things like that. You can buy enzymes. For instance, if you're lactose intolerant, you have a hard time digesting milk sugar, lactose. So they actually isolate lactases, enzymes that break down lactose, from certain fungi. So if you're lactose intolerant, you take some Lactaid. That's just lactase enzyme that's isolated from that are produced naturally by certain fungi. Keratinases are also popular. They're made by fungi and they digest keratin. And you think, well, it's used for a softening agent for certain clothes. Anyone want to guess? Well, if you've ever bought natural wool sweaters, you'll find out they're very itchy. So usually wool needs to be treated with keratinases to soften it so it's not as itchy and stiff. So keratinases make your sweaters a lot more comfortable. And finally, there are things like antibiotics, like you find in Neosporin here. And antibiotics are made again as a kind of defense. Microbes make them as defense to fight other microbes. Okay. A Neosporin has actually 3. We'll talk about that later, a few lectures from now. So what's the growth phase? So this is a primary. That's a primary. That's a primary. Vitamin, that's a primary. Enzyme, that's a primary. Antibiotic, no, that's a secondary. Another interesting product that's used a lot for organic farming is bioinsecticides. And there is a bacterium, Bacillus thuringiensis, and it makes a toxin that literally kills caterpillars, the ones that eat certain vegetables. And it kills them by forming pores in their GI tracts, in their guts. All right. And that results in the cells in the caterpillar lysing. They lose water and kills the caterpillars. It's all natural. Okay. So what's interesting is Bacillus thuringiensis is a gram positive. It makes spores. It sporulates. And for some reason that's not clear, they make these BT, Bacillus thuringiensis toxins, and they make crystals of them. So you can see here, there's things, and if you close up, these are the crystals. All right. So they're made during sporulation. So what kind of product is it? Primary or secondary? Secondary. Very good. Secondary. And for the product, what they do is they just grow up the bacteria. They grow them well at first, happily, and then they starve them. It causes them to form spores. They collect the spores, and that's what they spread then on plants that are sometimes eaten by the caterpillars. Well, the caterpillars, they end up eating the spores, which contain the Bt toxin, and that ends up killing them. Now, you can eat the spores, you can eat the Bt toxin, it doesn't bother your body at all. A little bit about these Bt toxins. As you could see in the picture before, they look like crystals, and the crystals are just filled with these proteins that are toxins. Okay, so they're actual protein toxins made by the Bacillus thuringiensis. And once it's consumed and gets into the body of a caterpillar, these toxic proteins form, which is pretty wild. They get in and they form these pores. All right, and these pores then form in the cells that line the digestive tract of the caterpillar. So these are cells of the caterpillar. And so what happens is water basically goes into the cells, ATP leaks out, and the cells of the caterpillars blow up and they just die. So now let's switch gears and let's go to recombinant products, or what we call genetically engineered recombinant products. And these are achieved through two major steps. Number one, genes are made or modified using things like you've heard about before: restriction enzymes, ligase, polymerase chain reaction, stuff like that, other technology. So they're made, and then they're sent into a microbe by recombination, right? Because recombination is the acceptance of DNA from an outside source. So some type of recombination is used to deliver the gene, usually transformation. So let me start with a little bit story of my childhood. When I was in college, like you folks, back in that year, yeah, I worked at a summer camp called Bearskin Meadow Diabetic Youth Camp. And when I started my freshman year working there, those are some of the kids that were at the camp many years ago. The kids there would be injected with insulin. They were diabetics. Their body could not make enough insulin. And the insulin was from cows. So it was beef insulin. Right? By the time I was a senior in college and working there, 3 years later, most of those kids were being injected with humulin. Humulin is human insulin that has been made recombinantly in bacteria. So within 3 years, the source of insulin went from naturally cows to being made recombinantly in bacteria. Why did that happen? What were the advantages? How did it happen? Well, just first of all, the how. Let's look at the big picture. What you need to do recombinant production of human insulin is the human insulin gene needs to be isolated from the human cell. It is put into a plasmid. So now we call this recombinant DNA because it's going to be used in a recombination reaction. It's going to be put into a microbe. So the microbe, which is recombinant or transgenic, now contains this plasmid with the insulin gene. And the bacteria now produce the human insulin protein. This is just kind of a big picture. I'll kind of fill in the spaces here. Now, that's the how. Why? Why would recombinant be better than natural? I mean, isn't natural always better? You know, that's what it says on the internet. The internet never lies, right? Well, number one, let's just kind of thinking back to if you're getting it from a cow's body injected into you versus if you're getting it from bacteria. There are a lot of people that are allergic to cow beef products. So some of the stuff from cow's beef will get into that insulin. And if you're allergic, you know, you might have a reaction to it. If it's being made in bacteria, there's less chance of that problem. It's much easier to purify and clean it up. So that's one reason you'd want recombinant. The second thing is, if you're getting it from these cows, all right, remember these cows are just cows were raised in field stockyards, and they could have had a lot of diseases, all right. They don't like make sure only certain cows were used. They pretty much used the pancreases of any cow that they had slaughtered. So sometimes, unbeknownst to the farmer, the cow was sick. It actually was infected with viruses and other disease agents. And those could actually get into the insulin that was purified from the cows. So that's another concern. Thirdly, and it's a big one, it ends up being a lot cheaper to make insulin, human insulin bacteria, than it is to isolate it from cows. And here's the big idea. Once you have something recombinant, you have a recombinant gene, it's in a plasmid. You, with that DNA in hand, you can change it. You can change the DNA so that the protein that is made is different. You can change, let's think DNA that's in the coding sequence, and that's going to end up causing change in the mRNA, change in the actual protein. So change amino acids. And if you change certain amino acids, sometimes you can make that protein work better, improve its activity. You're playing the creator now. You're making it better. Think about just causing some missense mutations in a protein. Sometimes the activity doesn't change in the protein. Sometimes it goes down, but sometimes it gets better. When you're using recombinant technology, you have the gene, you isolate it, you've got it, you can change, it's in a tube, you can change it. And then you put in that bacteria, and the bacteria will make the protein you program it to make. On the other hand, when you're getting protein from a cow, you can't go into the cow and change its DNA very easily. Now, there are some people who do that, but it's very, very, very difficult. All right, very, very difficult, very, very much expensive. So you really can't do it. What you get out of the cow is what you get. You really can't change the product, but you can change, modify the product that you're making in a bacterium. So enough about the why. Let's talk about the how. And should be a review for most of you, because we kind of went over this in the last couple weeks. To manipulate known genes to create new ones, you basically have to change the function of certain parts. And all y'all should remember that every gene has basically three parts, right? A promoter, a coding sequence, transcribed region, and then a transcription terminator. The promoter, again, is that switch for transcription. It controls how much mRNA is going to be made, so therefore how much protein will be made. If there's more transcription, there's more mRNA, and therefore there should be more protein made. The transcribed region, remember, contains the 5' UTR, the coding sequence, and the 3' UTR. And so remember, the 5 and the 3' UTR control how well the mRNA is translated, how many ribosomes bind to it, so how much protein can be made from every mRNA. And the coding sequence, of course, has the information for what amino acids go in here, and that controls what the protein does. And then there's the transcription terminator, our final one, and that's basically a region that tells the RNA polymerase, hey, stop transcribing, and it makes sure that the mRNA has a nice mature end. Okay, they're all very important, and they all can be manipulated as needed. If you look at an expression plasmid, that's a plasmid that's used to express proteins in a bacterial host. You see it has those parts, right? You have a promoter, the transcribe region, 5' UTR, coding sequence superior, and you have a transcription terminator. All right, now going back to transformation, okay, this can be recombinant. It's usually sent in by transformation. You need an origin of replication on the plasmid. So once it gets in the first bacterium, it will be copied. So all the daughter cells will get a copy of it. And remember, there has to be a selectable marker, a reason to force the bacteria to keep that plasmid around. Because if there's no reason to keep it around, it's a hindrance. The bacteria will want to get rid of it. Now, what I want to note here is we're using a bacterial host, so E. coli, and we're going to use it to make the human insulin protein. So we're sending this plasmid into bacteria. That's the host. Notice it's a bacterial promoter, bacterial 5' UTR, bacterial 3' UTR, bacterial transcription terminator. All right. Why is that true? Because remember, the host controls everything. The host controls everything. So the expression plasmid is put into a host. It's the organism that is going to be in charge of reading the information in the plasmid, following the directions, and making that protein. Now, think if you're going to visit a foreign country and you're staying in the host's home, everything will be in the language of the host. So let's say you're in Russia, everything's going to be in the Russian language. For you to survive, to be happy, you've got to be able to speak the language, understand the language. Well, the same way, the plasma that goes in there, all right, it has got to be able to communicate with the host. It's got to communicate with the bacteria. That's why we say that the host, the bacteria, is going to express that foreign recombinant gene. It's going to transcribe, translate, fold that protein. All right? So for that communication to work, the control elements of the plasmid, that's things like the promoter, the 5', 3', the transcription terminator, those have to be of the host. Because the host, the bacterial proteins in the cell, they're going to follow the directions. They need to be able to communicate with the plasmid. The plasmid has to speak their language. Now you say, well, why can't I put a human promoter? Why does it have to be a bacterial promoter? Why can't I use the original human insulin promoter? Because the DNA of bacterial and human promoters are very different. Why? Because the RNA polymerases in human beings and the RNA polymerases in bacteria are very different. All right? In humans versus bacteria. So again, the host controls things. It has to be in the language of the host. The only part of the expression plasmid that's the actual foreign gene is the coding sequence, right? The insulin coding sequence. Why? Because of the universal code, right? AUG means methionine from humans to bacteria. So that you don't have to worry about. You can leave that as the human. But everything else of the gene has to be that of the host. So let's talk about some tools of recombinant DNA, for instance, for making human proteins like insulin, E. coli. Right? You have to make the plasmid, that plasmid I showed you. How do you make it? Well, basically restriction enzymes, modifying enzymes like ligase, they're going to move around and stick together pieces of DNA in the circle thing called a plasmid. So this moving around of big pieces of DNA is known as subcloning. Right? That's subcloning. Moving around large regions of DNA. The second tool, which I'll talk about, is site-directed mutagenesis. And that is not moving around pieces of DNA. That is literally going to a piece of DNA that isn't something and making a small change in it, in the place where it already is. All right? Maybe changing a codon or changing a few nucleotides. Specifically changing them from one thing to another, changing an A to a G, removing 3 Gs, or adding 2 A's. That's site-directed mutagenesis. Okay? See the difference? Hopefully you do. Now, both of these require some type of vector plasmid, okay? So that's we kind of got to look at those. So let's kind of work with a plasmid here. And let's say we're going to make an expression plasmid to make human insulin in a bacterial host cell. What is involved? Here's a very simplified thing on subcloning, because we're moving pieces of DNA around and sticking them together, subcloning. So here we have a plasmid, okay? It's got a promoter, 5' UTR. It doesn't have a coding sequence. It just has a restriction site, restriction enzyme site, 3' UTR, transcription terminator. What's missing on this plasmid? If it's going to go into bacteria, it also needs, let's put it in there, an ORI and a selectable marker, ampicillin resistance, for instance. All right, what do you got to do? We got to move this coding sequence into here. We got to get to stick and insert that. Well, in order to do that, you have to have sticky ends to the DNA. So there's an EcoR1 site that is in this plasmid. If you cut it with the restriction enzyme EcoR1, you cut it to really create these sticky ends, so that here you cut it here, you cut it here, you cut between the G and the A and both, and it opens up, and now you've got these sticky ends. You also have the coding sequence of the human insulin gene. Just go along with me now. It has the EcoR1 sites at the ends of the coding sequence on the far edges. It's cut with EcoR1 restriction enzyme. It generates the same sticky ends. So notice these sticky ends, right? See, they're complementary. They can base pair. They can go in and base pair together. All right? So that's kind of what you want to think about. So these sticky ends, you're like, okay, I got these base pairs, but what makes them stay there as sticky ends? Okay? Because you could say this is going to come in here and it's going to base pair, but that's just hydrogen bonds. That's why, as it says down here, you need to use the enzyme ligase, and ligase seals them in there. It puts in covalent bonds in those restriction fragments so that they're permanently stuck together. Ligase seals the deal in those sticky ends. So here's another way of looking at it. All right, let's go. We have the insulin gene. Okay, it was cut with restriction enzymes. We use the same restriction on a plasmid. So now we have sticky, compatible, cohesive sticky ends. They're complementary to each other. They will base pair. All right. Then you add DNA ligase to seal the deal. Make it a permanent covalent. And now you do transformation, some type of recombination. So first, all these things here, the isolation, the cutting, the putting them together, the ligase to seal the deal, that's all done in a tube. That's all done in a tube with the purified enzymes, the purified DNAs. Once you have them in a tube, you then transform them by transformation process, uptake of naked DNA from the environment. You'll learn about this in a Labster lab coming soon. And then you basically transform the plasmid into the bacteria. The bacteria will pick it up. There's a selectable marker. So you'll grow the bacteria, for instance, on ampicillin, because the plasmid has an ampicillin resistance gene. The bacteria will keep that plasmid around. They'll do fission. So each parent will divide into daughter cells. And you have an ORI to make sure the plasmid's copied, a selectable marker to make sure the plasmid's kept around. The bacteria will then express the gene, the insulin gene that's in that expression plasmid. It'll transcribe it, translate it, and here in this example, export the insulin out into the medium to be purified. So that kind of gives you the big picture and all the steps, all right, of how to go from just pieces of pure DNA in a tube, cutting it, sticking them together in a tube, and then transforming it into the bacterial cells, growing up the bacterial cells so that they make the insulin. Here's just, you say, okay, I've got these bacterial cells. I want to do a large scale production. So I've got the bacteria. You put it in this. This is a large fermenter. Maybe this is, we're talking 100 to 1,000 liters. Okay. You clean the fermenter first. You make the medium. You sterilize the medium. That's a preparatory step. You then add the bacterium that can produce the insulin in there. You grow it under selection, okay? That way, because if there's no if there's no selection, like ampicillin in the medium, the bacteria will not keep the plasmid that has the ampicillin resistance gene. All right? So to keep the plasmid around, the plasmid has a promoter and all the parts you need for expression of the gene. All right? It is expressed. The insulin is made all during the fermentation step. The conditions are set so the bacteria make a lot of that insulin. And then you purify the insulin from the medium, and that's known as the third, which is the recovery step, right? Recovery step. So we're just recapping those three major steps that we talked about before for industrial fermentation. Okay. So that's about subcloning. Now a real quick thing about site-directed mutagenesis. In site-directed mutagenesis, you're knowingly changing. You know what you're changing. You're specifically changing a small, specific piece of DNA, usually in a plasmid, and you're changing it in a certain way to get the result you want. All right. Here's just an example of the result of site-directed mutagenesis. I don't want you to understand how the DNA was changed. I just want you to realize, oh yeah, the DNA was changed, and that led to the change in the protein. So let's kind of get to here. You originally start with a plasmid, okay? It is encoding for a protein, and here's a codon, GGG. That's, here's your codon, GGG. Now notice, hold it, it should be GAG, but site-directed mutagenesis was done. I don't care how you did it, just know it was done. So instead of GAG, the plasmid says GGG. All right. So again, this is one in one cell. Okay. The strands are used then as parents. You're gonna get two plasmids made. All right. One plasmid will have the new GGG, CCC, okay, with a mutated codon. The other will just have the original plasmid sequence because this lower strand went to this cell, so we have the GAG, okay, which is okay. So they're put in bacteria. Here, the bacterium that has the original gene, okay, it makes a protein, GAG with leucine. Okay, so that GAG encoded the leucine. Here's your natural protein. On the other hand, the bacterium that got the plasmid with the site-directed mutation, the specific change of GAG to GGG, GGG will encode a proline. So now the bacteria with this plasmid makes the same protein, except instead of a leucine, there's a proline in that protein. And ideally, this change will make it a better protein. All right. For hirudin is a protein. It's originally from leeches. This is a little cocktail. It's originally from leeches. The gene was isolated. It was put in a plasmid, and hirudin was expressed in a bacteria. The hirudin basically is an anticoagulant, okay? It stops blood clotting. They did this one mutation, so this is kind of a missense mutation if you think about it. You just changed one codon, you've substituted one amino acid for the other. And they were very fortunate because This led to a 300% increase in the activity of hirudin. So the original hirudin, the one with the leucine in it, it had good activity. That was in nature. But if you changed one amino, the right one, and it was the right amino acid, in this example, and this is a pretty extreme example, but it is a documented example, one amino acid change, one site-directed mutation increased the protein 300% activity. Okay, that's pretty impressive, but it shows you the potential of recombinant DNA where you are the creator. You can improve on nature. So at this point, let's talk about some amazing examples of recombinant DNA technology. Okay, and I'm going to go into some specific examples here. All right. Number one is biosensors. All right. This is actually used when you go to sewer plants and things like that. Okay. This is a subcloning application. All right. Where they basically moved a gene from its original place into a plasmid and put the plasmid in the bacteria, and the bacteria made the protein. Okay. This is a subcloning application. The purpose is to create microbes that can sense whether toxic products are present in wastewater, sewage water. In this example, a toxic product is like a pesticide, a herbicide, maybe a chemical that has cancer-causing potential. All right. I want to start with an analogy of a canary in a coal mine. I don't know if everyone's heard of this, but in the olden days when you had coal miners and they were mining coal from deep in the earth in these caves, sometimes there were poisonous gases in these caves. The coal miners would go there. The poisonous gases were odorless. They'd breathe them in and they would die. So what they decided to do was, in order to save the coal miners' lives, they would send canaries, birds that sang, into these caves first, into these mines first. If the canary kept singing, that means the canary was alive and it was fine. And they would send the coal miners in. But if they threw the canary in there and it stopped singing, that told them, uh-oh, that canary's dead. Don't send the coal miners in there. So you could imagine the canaries in the coal mine were there to save the coal miners. In the same way, biosensor bacteria are used to save the lives of human beings. Okay, how is that so? Okay, so you're using microbes to detect toxic products. Now, in wastewater has poop in it. Poop is not considered toxic in this example. We know poop's in there. That's okay. We want to find out, is there something like a cancer-causing pesticide or herbicide or some other contaminant in there? That's what we're testing for. Okay? Because that chemical would hurt humans, but would also hurt these microbes. So the microbes are canaries in the coal mines for humans. All right, so let's start with Photobacterium. Members of this genus have what's called a Lux gene. The Lux gene encodes a Lux protein. It is an enzyme that triggers the emission of light. So here's a fish. It has some photobacteria under its eye. The bacteria are growing on the fish under its eye. They are expressing Lux protein and they're basically emitting light. All right. Now, these photobacterium, they live in fresh water. They grow very well in fresh water. They make lots of light. But if you put them in wastewater, they would die. Okay? They would die in wastewater. Why? Because they don't like poop. So the solution, though, what they did is this. They said, well, E. coli like to live in wastewater and poop, but they don't emit light. So let's take the best of both worlds. Look, Miley Cyrus again. Their goal was to engineer recombinant DNA E. coli that contain the Lux gene from Photobacterium so that these E. coli would make the Lux protein
and therefore emit light. That's the goal. Okay? So, based on what I've told you right now, this shouldn't be too hard to understand how they did it. They basically made a plasmid. They had the E. coli constitutive promoter, E. coli 5' UTR, the Photobacterium lux coding sequence, the E. coli 3' UTR, and E. coli transcription terminator. Those were all put together by using restriction enzymes and ligase, by subcloning them, all right, into a plasmid. And then they transformed this plasmid into E. coli. E. coli is the host. That's why you had to use E. coli promoter, E. coli 5' UTR, and so forth. It's the host. Remember, there had to be an ORI and a selectable marker too on the plasmid for maintenance. Now, what meant was the Lux gene, therefore, was transcribed and translated constitutively. Okay? Well, why? Why? Here I said constitutive promoter. What does that mean? A constitutive promoter is one that is on all the time at a very high level. So a constitutive promoter would make sure that the RNA is transcribed at a high rate, high level, all the time. So that way the Lux gene was transcribed into mRNA. The mRNA was translated constitutively by the bacteria. So the bacteria was making this protein and ideally emitting light all the time. Okay? And remember, the Lux enzyme produced that light. Okay? It's the Lux enzyme that's producing that light. So let's look in this example here. All right. Here were the bacteria, all right, that were recombinant. They're producing Lux enzyme. They're put in wastewater, all right, different batches of wastewater. Look, they're growing fine. They're emitting light. Great, great. But hold it, as there is more toxin, herbicide, pesticide, whatever, what happened? The bacteria start dying off. They start making less light. And it tells you, oh, the bacteria are dying off, right? So from just looking at these samples, you can tell that these later samples must have toxin in them. That's why there's less light there. So the bacteria did not grow well. If they don't grow well, they don't make as much light. And so it's very easy to tell just by looking at these bacteria in these different water if the water's safe or not. If it failed to emit light, that tells you, of course, that the toxin is present at a really high level. Probably a level that if humans consumed it, it would hurt them. Now, since the original work, okay, they improved the Lux protein. How? They did site-directed mutagenesis, SDM. So they made changes in the plasmid, in the coding sequence of the Lux, coding sequence of the Lux gene, missense mutations. So they swapped out certain amino acids for others. And in doing that, they created a Lux enzyme that was more efficient. And therefore, when the bacteria made it, they went from pretty fluorescent to a lot more fluorescent. Okay, so greater sensitivity, and that was helpful in doing this water analysis. Now, there have been problems using recombinant DNA. The first one is brought out by this cartoon on the left, where you see these recombinant microbes, P. putida, say, oh dear, I didn't realize in the field it would be like this. We should have stayed in the laboratory. So, and this points out that a lot of times these genetically engineered microbes, they don't survive. They don't compete very well when they're put in the wild, because in the lab, a lot of things have been done to them. DNA has been added, DNA has been taken away, and they're just not as hardy anymore. So when they do get put in the wild, they get eaten or die. For instance, the recombinant E. coli that I talked about, which is kind of a biosensor, it's had problems because sometimes you put it in the wastewater and it just can't compete and it dies off, so it has to be constantly added over and over again. So that's one problem. Number two is a perception problem.
on the right here, you see Hungry Man, Franken Food, Frozen Dinner, Monsanto. So Monsanto is a company that makes uh, recombinant products. Okay, they've actually expressed different genes in different vegetables and things like that. And you'll hear of Wolfgang Puck and some other famous celebrities say, "Well, I'll never use genetically modified food," and stuff like that. So, is it this fear founded? Well, overall, probably not. There are some concerns, but potatoes with eyes, tomatoes with fish. I mean, tomatoes with fish tails. That's a little far fetched. But again, there's a perception that these things are dangerous. So it's fine to have recombinant microbes make insulin, which is injected right in the bloodstream, but you don't want recombinant microbes in food, which also go into your body. But that's just the way it is. I'll just give you an interesting story. In UC Davis had a very good wine program there, and they actually make recombinant yeasts, and these recombinant yeasts actually have abilities to make pretty good wine. Uh, so in some cases, wine that's better than things that natural yeast can make. However, they're not used in production because of the fear that if the public heard that, oh, wine was made with genetically modified yeast, most people would say, that's dangerous. I won't drink that wine, and that would ruin the company. So, because of that, a lot of genetically modified yeasts, which could possibly make really good wine, they'll never make it onto the open market. That wine might be made and studied in a lab, but it'll never get out there. Okay, so I'll just leave that at that. Give you something to think about. Good luck. Good day. Take care of yourself and be healthy. Bye bye.